Hi everyone. In this lecture, we're going to learn about DNA processes. So you're basically going to look at what happens to DNA in your cells and bacterial cells. Basically, why do we even have DNA? And we're also going to talk about how we can use microbial DNA for our own benefit. And that's the whole field of biotechnology, which we'll learn about more later, but we really have to understand how DNA is used in cells so that we can manipulate it for our own use in medicine, in genetics, and so many different things. So that's what this lecture is going to focus on. And we're going to talk about the ways that DNA can be studied to understand various microbial diseases, how antibiotic resistance develops in microbes, which is basically microbes gaining DNA that they didn't have before. And then again, how we can use this in medicine. So the lecture of this outline, we're going to start off by talking about the structure of DNA and RNA, so these nucleic acids. Previously, we learned that nucleic acids are macromolecules that are found in cells, and DNA is found in all different types of cells. So every cell in the world has DNA, and all cells also have RNA. So we're going to learn about the structure of these molecules so we can further understand how we can use them in the future with technology and genetics. And then we're going to learn about DNA replication, protein synthesis, the differences between protein synthesis in prokaryotic and eukaryotic cells, and gene regulation. And I'm going to break this lecture into two parts because it is a lot of information. I also just want to say right now that there's been a lot of work on a vaccine for COVID-19, so for SARS-CoV-2, which is the coronavirus that causes COVID-19. And there has been two areas of research. One area of research is working on creating a DNA vaccine, and another area of research is working on an RNA vaccine. So once we finish these lectures and you know all about this, we can discuss that further. But I want you to really understand the structure of these molecules so you can understand why one vaccine may be better than the other, or vice versa. Maybe both are great. So it's still a big area of research. And this morning, by the way, the FDA did stop research in a big biotech company in the Bay Area because they were working on vaccines that are DNA-based vaccines. And DNA-based vaccines can be a little bit more problematic than RNA-based vaccines. So I'll talk about it later once we get through the lecture and you all learn about these molecules. Okay, so to set the big picture, kind of like what we were talking about, understanding genes and DNA mean the same thing. So when someone says, I'm looking at a gene, it means that they're looking at a DNA. Understanding bacterial genes is important. And here are some reasons why it's important. So many diseases are caused by microbes. If we can understand what it is that microbes are expressing, what genes they're expressing, or what DNA they're making, we can create drugs that can target those genes so that we can target those microbes. We can also, we have a big problem right now with microbes that are antibiotic resistant. What that means is you have a microbe and an antibiotic used to work on it in the past. So someone has an infection, could be any infection, could be like a respiratory infection, a digestive infection, a urinary infection, and they have, let's say they have a bacterial infection, and you give them antibiotics, which is great. In the past, a lot of our antibiotics used to work very effectively, but right now they're not that effective. There's also many research studies that are coming out that are saying in the days of COVID, so this year, because we have so many people going to the hospital for COVID-19, and remember, COVID-19 is caused by a virus, not bacteria. They're different things. A patient comes into the hospital and they have COVID-19, but because their immune system is compromised, that's what immunocompromised means. So they're weak, they end up catching a lot of bacterial infections while being at a hospital. And then we end up giving so many antibiotics to that patient that right now, not only are we seeing a rise in COVID-19 cases, we're seeing a rise in antibiotic resistant bugs because of COVID-19 and all the antibiotics we're giving, again, for bacterial infections, not COVID-19, but because a person is immunocompromised when they have the viral infection, a lot of doctors worry that they will get so many bacterial infections. So as a precautionary measure, they'll sometimes end up taking antibiotics. So just keep that in mind. And then biotechnology. So all the biotechnology that you hear about in different companies in the Bay Area working on the newest drugs or the newest chemicals that we can use for cars or for farming or anything like that is all done through biotech. And that's basically altering genetics for our own benefit. So an example of where biotech has been very useful is in insulin. And most of you know of a diabetic person, or you yourself may be diabetic. So a lot of people who are diabetic take insulin, and we created insulin by using bacteria. And we'll talk about that later. So hopefully this set the stage for understanding why these things are important, because the beginning of it is a little bit boring, but I do really want you to understand the structure of these molecules so that in the future, when we talk about the more detailed fun stuff, you understand what I'm talking about. So DNA and RNA are nucleic acid molecules, and they store genetic information of all organisms. The two DNA processes that we're going to talk about replication. So one part of this lecture, which is today's lecture, we're going to focus on replication. Replication is the DNA molecule making a copy of itself. So DNA making DNA. And the reason why this is important to understand this process is when mutations happen or things go wrong, this can really affect a cell. And this can also, if we're talking about human cells, for example, that's what can lead to cancer. So many mutations happening. So we're going to talk about how DNA makes a copy of itself. And remember, with microbial cells, before they divide, they do binary fission. So they have to make a copy of their DNA before the bacterial cell can divide into two cells. And then the next part of this lecture will be about gene expression, or how your genes, your DNA is expressed. And that's also called protein synthesis. So to your DNA, your genes are important because they code for protein, and protein makes us who we are. So we're going to talk about how DNA codes for proteins, and we're going to talk about the process of DNA getting converted to RNA and RNA getting converted to protein. Now, this is the first time we really talk about RNA. So RNA is kind of the middleman in this process. So that's where we're going to mention RNA. But the first slides are going to focus on the structure of DNA and RNA before we get to these processes. Okay, so the structure. I want everyone to know the structure of DNA and RNA. So DNA and RNA are made up of nucleotides. Remember in the macromolecules lecture, I said the monomers of DNA and RNA are nucleotides. And when we look at a nucleotide, whether it's a DNA nucleotide or an RNA nucleotide, there's always three components. Every nucleotide has a phosphate, it has a sugar, and it has a base. So the sugar in DNA is deoxyribose, and the sugar in RNA is ribose. Every nucleotide has a nitrogenous base. DNA comes with 4 different nitrogenous bases. There's adenine, which sometimes we just say A, thymine T, cytosine C, guanine G. And keep in mind that DNA has thymine, which is T. RNA has adenine, uracil, cytosine, and guanine. So these are the nitrogenous bases that we see in uracil. So again, the nucleotide structure of both DNA and RNA, there's a phosphate group, there's a sugar, it's deoxyribose in DNA, it's ribose in RNA, and there is a base. And the main difference is that in DNA we see thymine, and in RNA we see uracil. And one other thing I want to say is when we look at the bonding between nitrogenous bases, which I'll talk about in a second, adenine always binds to thymine. So A always binds to T in DNA, and C always binds to G. In RNA, A always binds to U, and cytosine C always binds to guanine G. So DNA and RNA, they're sugar phosphate backbone molecules, meaning that when you look at the molecule, the sides of the molecule are made up of phosphate and sugar groups, and then the nitrogenous bases are in the middle. And then the nitrogenous bases are hydrogen bonded to each other. So again, A binds to T, and I know this is DNA because there's T here, and then C always binds to G. Okay, and then the order of the the nitrogenous bases makes you who you are. So whenever you hear of people getting their DNA sequenced, basically you send a saliva sample to these companies like 23andMe and Ancestry, and what they do is they do some work in lab to extract the DNA from your cells. So they open up the cell membranes, and then we're eukaryotic, so they open up the nucleus and they get the DNA, and then they basically pipette or stick the DNA in a machine, and the machine tells them the order of nitrogenous bases, and you get thousands of nitrogenous bases based on the organism. So maybe if I were to stick my DNA, it would be like ATTCGAT. It would just be a random order. This doesn't mean anything, but this makes me who I am. So if I compare my genetic sequence to maybe 1,000 other DNA sequences, I might have genetic sequences that are more similar to people who are from various parts of the world. So that's how Ancestry is figuring out where people come from. They see that, okay, the DNA from maybe China, India, the Middle East, Japan, all these different countries, all the countries in Africa, you see a similarity of DNA generally with the people there. So that's how they're figuring out Ancestry. And the more people that get sequenced, the more we learn about them. We can also look at individuals, for example, who have diabetes versus those that don't, or people who have a certain type of cancer versus those that don't. And we could see that there are some similarities and differences in their DNA. So the nitrogenous bases order is really important because it makes us who we are. Okay, DNA, just to summarize, it's double-stranded. It has complementary strands. So adenine always binds to thymine, cytosine always binds to guanine. So adenine is hydrogen binded to thymine, and cytosine is binded to guanine. The strands are anti-parallel. I will talk about what that is in a second, but so far I want you to know that. And then we know that DNA has thymine, which we do not see in RNA, and the sugar is deoxyribose. RNA, there's 3 different types of RNA. Remember, we're going to talk about RNA later, but RNA is basically the middleman between DNA getting converted to protein. So there's 3 types of RNA. There's mRNA, which stands for messenger RNA, rRNA, which is ribosomal RNA, which is what we see in the ribosome, and tRNA, which we will learn about later, which stands for transfer RNA. RNA is single-stranded. It has uracil instead of thymine. It does not have thymine, and uracil binds to adenine. The sugar is ribose and shorter than DNA. Now, I want to say something important. If you look at basically any biology or microbiology textbook, most of the time they'll tell you DNA is double-stranded and RNA is single-stranded, but I do want to point out that this is the general case most of the time, but DNA can be found single-stranded and RNA can be found double-stranded. So research has shown this, but this is the textbook definition of these things. Now, if I were to ask you, what is complementary to these bases, U, A, G, C, A? U, the complement to that is A. For A, it's U. For G, it's C. For C, it's G. And for A, it's U. And is this DNA or RNA? I know it's RNA because there's a U and there's no uracil in RNA. And here's a good video that explains this.
DNA and RNA are nucleic acids, polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has 1 more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered, beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group.
Okay, this is very important. So whenever, some of you have already learned this in the past, but if in the future you see me say 5' and 3', what these numbers refer to are these carbons. So I'm looking right here at a DNA molecule right here, and here's RNA. Anyway, remember that for DNA, for the nucleotide, there's a phosphate, the sugar, and then a nitrogenous base. And for DNA, that can be adenine, thymine, guanine, or cytosine. And then for uracil, we have our phosphate group, our ribose, sugar, and then the nitrogenous base, which could be adenine, uracil, cytosine, and guanine. Now, the important thing is in this video, he talked about the numbering of carbons on the sugar. Whether you're talking about deoxyribose or ribose, the carbons are numbered. The carbon binded to the nitrogenous base is 1', 2', 3', 4', 5'. So the 3' carbon, whether it's on DNA or RNA, here's the 3' carbon, that's where the next nucleotide gets attached. So whenever we have a new nucleotide coming, it comes to the 3' end. So when we say DNA grows 5' to 3', we mean that you're adding a nucleotide on the 3' end. And that is the 5' end is where the phosphate group is. So just to make that
clear. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide.
So now whenever you hear
RNA usually consists of a single...
So the main thing I want you to get is now whenever you hear me say that DNA and RNA grow from 5' to 3', it means that at the 3' end is where we're adding the next nucleotides. The 5' end is just the end where we have the phosphate group. I'm going to go to the next slide. And then he basically says that RNA is generally single-stranded, DNA is double-stranded. In RNA, adenine binds to uracil, in DNA, adenine binds to thymine, and in both, cytosine binds to guanine. Okay, so now that we covered the different structures of both, it will probably be a test question. What are the differences between DNA and RNA? So I want you to cover those. Now we're going to talk about DNA replication. So we're going to put RNA aside. So we're going to talk about how does DNA make copies of itself. So how do you take one DNA molecule and make two DNA molecules? For microbes, for bacteria, this is important because, again, before bacteria divide or do binary fission, they may need to make a copy of their DNA. For human cells, this is important because, for example, when you hurt yourself, your skin cells need to make more cells. They need to make a copy of the DNA. So this is DNA replication, and all it is making a copy of the DNA. We're going to learn about this process. In bacteria, DNA replication happens right before bacteria divide or before binary fission. And the location where your DNA replication starts is called the origin of replication. DNA replication is bidirectional, means it goes in both directions when we're looking at how DNA is replicated. So you have your origin of replication, and then DNA is replicated on both sides. And DNA replication is also semi-conservative. Semi-conservative is a fancy word for saying that once replication is done, your two new strands of DNA, each one has one old strand and one new strand. So it's half conservative. So each new DNA strand has one old, which we call the parent strand, and one new strand, which we call the daughter strand. So here's a video of DNA replication, and then we're going to go over the process. And again, just to remind everyone, because I know a lot of this can sound very boring, memorizing all this stuff, it's really important to understand how DNA replication works, because I would say that for most diseases, something went wrong with DNA replication. Or for most microbes that cause really toxic diseases, it's because they create a new gene where some mutation happens. So it's really important to understand this process if we want to move the field of medicine forward. Okay, so here's a video.
Every cell in your body is produced by cell division. Before each cell divides, it must copy its genetic material in a process called DNA replication. Understanding of DNA replication comes largely from studies of E. coli, bacteria that are found by the billions in your large intestine. Let's take a look at how DNA replication occurs in an E. coli cell. As we zoom in, we see the DNA. At the origin of replication, the two strands of DNA separate.
So remember, I said the origin of replication is just a fancy word for saying where DNA replication actually starts. And the reason why it's important to know the origin of replication is some researchers, when they're studying how DNA replication happens in like a new bacteria that's causing disease, they go and look for the origin of replication. So now this narrator of this video is saying, first, here's the origin of replication, and where replication happens, that the sides of replication, we call it the replication fork, and that's where all the enzymes will come in.
As templates for making new strands. The result is a replication bubble. The bubble grows in both directions, forming two replication forks.
So that's what I meant when I said that replication is bidirectional.
Zoom in on one of them. Many proteins work together at the replication fork. Only some are shown. Here, the DNA is unwound, and DNA polymerases, shown in orange, build new strands of.
Remember, everything that ends in ASC, 99% of things are enzymes, so that's why she said many proteins are involved. She means many enzymes are involved because enzymes are proteins. So now what she's going to do is she's going to tell you all the enzymes that come in that help make DNA copy itself or replicate itself. And one of the very important enzymes is DNA polymerase. DNA polymerase is basically the main enzyme involved in DNA replication. It's the enzyme that makes new DNA.
Original parental DNA strands are shown in dark blue. Newly formed DNA strands are shown in light blue. Because strands in a DNA double helix run in opposite directions, the new strands must be made in different ways. One new strand, the leading strand, is built continuously. The other new strand, the lagging strand, is built in pieces.
Okay, so let me stop the video here. So earlier I mentioned that DNA is double-stranded. So there's two strands of DNA. Now remember, in bacteria, DNA is circular, but let's just imagine a circle and it's double-stranded. So there's one strand here and one strand here of DNA, and they're bound to each other by nitrogen bases. Remember, in the middle here, A is binding to T and C is binding to G. So they're bound to each other. So at the origin of replication, they open up a little, and then you want replication to happen. When we see the strands are anti-parallel, we mean that when you look at one strand, we see 5' to 3', and the other strand is 3' to 5'. So when DNA replication happens, in one strand is gonna make a copy of itself much faster than the other strand because the 3' end is already available for you to add nucleotides to. And that strand we call the leading strand. The other strand has a 5' end out, so we need to add a piece of 3' DNA, so we need to add a primer. So that is where the synthesis of the lagging strand happens.
First, let's focus on the leading strand. DNA polymerase builds a new strand of DNA by adding DNA nucleotides one at a time. Each new nucleotide must pair up with its complementary nucleotide on the parental strand. Adding new nucleotides works the same way on both the leading and lagging strands. Each piece of the lagging strand begins with a short segment of RNA, shown in red. A clamp surrounds the RNA and attaches to DNA polymerase, which builds the rest of the new piece as DNA. When the piece is finished, it is released from DNA polymerase. How are pieces of the lagging strand joined together? A different DNA polymerase removes RNA and replaces it with DNA.
Okay, so
However, it cannot finish connecting the pieces.
So I'm gonna move on with this video. It's not the most exciting video, but I just wanted to talk about these components and I'll discuss them in the next slides. Okay, so the way DNA replication happens, if I were to write it in order, so how DNA makes copies of itself is you have a lot of proteins involved or enzymes. And these are the enzymes I'm going to focus on that I want you to know. So there's an enzyme called gyrase or topoisomerase. And for purposes of this class, I'm going to say that they're very similar or they're the same type of enzyme. So DNA gyrases or DNA topoisomerases, their job is to relax supercoiling. DNA is very supercoiling, meaning it's very tightly bound on each other. If you're going to make a copy, you need to relax it a little. So DNA gyrase, DNA topoisomerase, whatever you want to call it, depending on the textbook you're working with, that enzyme will come and will relax the supercoiling. Once you relax the supercoiling, another enzyme comes called DNA helicase. DNA helicase will unwind the DNA helix. So remember, DNA is double-stranded, so helicase will come and unwind it. Now primase will come and put a piece of primer so that DNA polymerase can come and sit on it on the lagging strand. And DNA polymerase is the main, so the most important enzyme in all of these is DNA polymerase. DNA polymerase is the enzyme that's going to come and add complementary nucleotides and proofread when something goes wrong. So DNA polymerase, when it sees adenine, is going to add thymine. When it sees guanine, it's going to add cytosine. When it sees cytosine, it's going to add guanine. So C always binds to G, G binds to C, A binds to T, T binds to C. And then finally, DNA ligase is the enzyme that will join the fragments together because one strand is the leading strand and one strand is the lagging strand. And then in this image, this is from your textbook. It shows all the enzymes and the order of things involved, but this is the summary of what I want you to know. And then we'll talk about the lagging strand and the leading strand. Okay, so here is the DNA shown. And the main thing I want you to take from this is that it always replicates by adding nucleotides on the 3' end. So you can only add new bases on the 3' end. So that's why we say that DNA always grows from 5' to 3'. And so this is where you will add your new bases. So during DNA replication, when you have both strands like this, one strand will serve as the leading strand, which will be copied continuously, and one is the lagging strand. The leading strand only needs one RNA primer because it already has the 3' end out. The lagging strand has discontinuous replication because it has the 5' end out, and you can only add nucleotides at the 3' end. So we have to keep adding RNA primers. So the lagging strand grows much slower than the leading strand. So if you were watching a live video of this happen, you'll see that when gyrase comes, relaxes supercoiling, then helicase comes and unwinds the helix, then the RNA primer comes, one strand will replicate much faster than the other. The one that's slow is called the lagging strand, and with the lagging strand, we see many RNA primers. And the little fragments of DNA made are called Okazaki fragments because the Japanese scientist who discovered them is named Okazaki. So if you're watching more YouTube videos, you'll see a lot of people refer to those fragments as Okazaki fragments. Okay, this slide is not working, but I think it's just summarizing. So here.
In principle, copying DNA, a process called DNA replication, is very simple. The two complementary DNA strands separate, and because each nucleotide can only pair with its complement, adenine with thymine and cytosine with guanine, each strand can be used as a template to build a new complementary strand, producing 2 DNA molecules.
In the cell, DNA replication is a little more complicated, but the principle is the same. For clarity, we have untwisted the double helix. Remember that each DNA strand has a 3' and 5' end, and the strands run in opposite directions.
That's what it means when you're reading on my slides or in your textbook that the DNA strands are anti-parallel. They're not parallel. One has a 5' end out at one end and then the 3' end on the other end. And then the other strand is 5 to 3. So one strand is 5 to 3, the other strand is the other way, 5 to 3.
DNA replication begins at specific sites called origins of replication. Proteins attach here and separate the DNA strands, forming replication bubbles, which grow in both directions. Enzymes called DNA polymerases move along the template DNA strands and catalyze the elongation of new strands. Because DNA polymerases can only assemble new DNA in the 5' to 3' direction, only half of the new DNA can be synthesized in one continuous piece. The other half is synthesized in short pieces. As the replication bubbles grow, one daughter strand is synthesized continuously, while the other daughter strand is synthesized in pieces. The pieces are joined together by the enzyme DNA ligase. Eventually, all the replication bubbles merge, yielding two identical DNA molecules.
So he basically just showed the leading strand and the lagging strand. The lagging strand has a lot of RNA primers that need to be removed. And then in the end, you get two DNA molecules made from DNA replication. So to summarize DNA replication, DNA gyrase or topoisomerase is the first enzyme that comes to relax supercoiling. Then we need to unwind the helix, and that's DNA helicase. Then primase comes and puts an RNA primer for DNA polymerase to sit on. DNA polymerase is the enzyme that makes DNA, so we're making new DNA here. So it's the one that will add complementary nucleotides, and it will proofread. Proofread means that if it made a mistake to accidentally put C where it should have put A, it will go back and fix it, and we'll talk about proofreading later. And then we always have 5' to 3' growth of DNA because we only add nucleotides to the 3' end. DNA ligase will come in the end and join all the DNA fragments because on the lagging strand, you have so many Okazaki fragments because you had to remove all the RNA primers. So then you have to fill in those holes. And so DNA polymerase will come and fill those holes with nucleotides, and DNA ligase will come and join all those fragments together. So I'm going to end the lecture here. So this was DNA replication. In part 2 of this lecture, we'll talk about protein synthesis, but I wanted to separate them. So the first part is DNA replication, so take time understanding that. And then in the second part, we'll talk about how we use DNA to make protein. And that's where we will talk about RNA again. Just take time really understanding DNA replication.